DNA replication is a very important part of life and functioning. As a whole, if DNA were not able to replicate, cells would not be able to reproduce, or they would mutate. While these mutations may not be harmful, some could potentially be harmful or fatal. DNA replication is what gives us chromosomes, is what is in cell nuclei, and is what controls gene expression. Without these functions, people, parents, and their cells would be drastically different from each other. Additional ways you could learn why this topic is so important and what it is to research the mutations or disorders caused by replication errors, and more in-depth discussions of the steps of DNA replication to further learn exactly what they do and why they are so important.
In biology, whenever one DNA molecule produces two identical replicas of the same DNA molecule, that process is called DNA replication.
DNA replication is a process that occurs in all living organisms and is the platform for biological inheritance.
Replication begins at a location on the double helix known as ORC, to which certain initiator proteins bind and trigger unwinding. Enzymes known as helicases unwind the double helix by breaking the hydrogen bonds between complementary base pairs, while other proteins keep the single strands from rejoining. Certain proteins surround the unzipping strands and relax the twisting that might damage the unwinding DNA. The cell prepares for the next step, elongation, by creating short sequences of RNA called primers that provide a starting point of elongation.
Elongation is the step after initiation in the DNA replication process. During elongation, an enzyme called DNA polymerase adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strands. This template strand specifies which of the 4 DNA nucleotides is added at each position along the new chain. Elongation can get very complicated, but it's very important to remember your leading and lagging strands and also these two things. During elongation, a primer sequence is added with complementary RNA nucleotides, which are replaced with DNA nucleotides. And also, your leading strand is made continuously, while your lagging strand is made in pieces that are called Okazaki fragments.
In the DNA replication process, the addition of telomerase assists the cell by protecting it. It is added to the end of the chromosome. In addition, it is done by an enzyme. The telomerase helps keep the cell alive. As the cell division occurs, this telomere shortens and also experiences a small loss of DNA. The telomerase help keep the chromosomes from being damaged and dying. A side fact, cancer cells typically contain more telomerase than regular cells, is a reason why it's difficult to get rid of the already uncontrollably multiplying cancer cells.